Hi, Shinya. Please say hi to Naoko for me too, and you should be emailing me some baby pictures soon. I don't have time to prepare a presentation, but I'll be more than happy to read these slides to you. And it might be a little long for 6 minutes. I don't know where you can shave some stuff, or you can just talk really fast. I kind of went through it and it took me about 10 and I talk fast, but you're also intimately more familiar with this data than I am. But yeah, hope you're doing well, and I'm basically just going to read these slides to you and I hope it helps. For your introduction slide, your title, hyaluronan synthase 2 overexpression inhibits activated matrix metalloproteinase 13 expression in human chondrocytes. And a little bit of an introduction. Hyaluronan, abbreviated HA throughout this presentation, is a major component of the extracellular matrix. It plays a role in the retention of cartilage proteoglycan and cell signaling. It is made by a synthase, abbreviated HAS2, so hyaluronan synthase 2, and that is the predominant isoform involved in the synthesis of hyaluronan in human cartilage. And there are 3 isoforms, and they're HOS1, 2, and 3. In the image below, you can see a chondrocyte that expresses HOS1, HOS2, and HOS3, making their hyaluronan strands outside the cell. Here you can also see CD44, which is the receptor for hyaluronan. And when hyaluronan is bound to CD44, your proteoglycan molecules can align on its surface. The next slide. Previous studies have shown that proteoglycan is lost during the development of osteoarthritis in cartilage. And here we show that by the loss of saffron and O staining and the lack of biotin hyaluronan binding protein staining. So the control, nice bright red staining. When you add interleukin-1, IL-1 alpha, which is our experimental model for osteoarthritis, you can see a loss of red staining in the cartilage. And you can also see a loss here, which I think that's a Western blot over here to the right. Hyaluronan injection is widely used for patients with knee osteoarthritis. And several studies have shown the benefits of hyaluronan on knee osteoarthritis. So clinical studies have shown a decrease in pain and effusion with an increase in function. And some basic scientific studies have shown a decrease in inflammatory factors, so the MMPs, interleukins, nitric oxide, prostaglandins, and TNF-alpha. However, little is known about the effects of endogenous hyaluronan on osteoarthritic chondrocytes. And so that brings us to 2 questions. Can we enhance the endogenous hyaluronan production by HOS2 overexpression? And does this overexpression reverse the progressive pro-catabolic effects of osteoarthritis? So the objective here was to examine the effect of overexpression of HOS2 on MMP13 expression in human chondrocytes. So we're using MMP13 as our marker for inflammation of osteoarthritis. We used human osteoarthritic chondrocytes for cell culture, and they were obtained from articular cartilage of patients who had undergone total knee arthroplasty. The activation of MMP13 was used as our endpoint, and we can increase the expression of MMP13 with treatment with interleukin-1 beta. Let me do that again. The addition of IL-1 is our model for inducing osteoarthritis in the lab, and which is shown to induce MMP13 expression. So we also looked at other inflammatory factors, TNF-alpha, lipopolysaccharides, and hyaluronan oligosaccharides, and all of these also induce the expression of MMP13. Similar results with these other inflammatory factors were found, but are not shown today. We used an adenovirus that was created with PCR that had a murine HOS2 coding sequence inserted, and it also had a MIC tag for identification. We used an adenovirus with a LAC-Z with no doxycycline as our control. So this is how we introduced the HOS2 gene into our chondrocytes. We determined the size of hyaluronan with an agarose gel assay, and we determined the quantity of hyaluronan using an ELISA. We analyzed our gene expression with QRT-PCR. GAPDH was used to normalize our mRNA expression. We looked at protein expression by Western blotting and used beta-actin as our reference. Our statistical analysis was performed with a two-tailed unpaired students t-test for direct comparison of treatment group to control. For multiple comparisons of groups, a one-way or two-way ANOVA followed by a Tukey Kramer's test or Dunnett's multiple comparison test were used. Data shown as mean plus or minus standard deviation, which are represented by error bars. And so now on to our results. To examine if hyaluronan is increased by HOS2 overexpression, our chondrocytes were transduced with the adeno-HOS2 virus and treated with doxycycline. Hyaluronan gel assay shows that the hyaluronan amount increased in proportion to doxycycline concentration. So the more doxycycline, the more HOS2 expression. In addition, the hyaluronan was high molecular weight as compared to standards. Next, chondrocytes transduced with HOS2 were treated with IL-1 beta, with or without doxycycline. A significant increase of MMP13 was observed with IL-1 beta alone as expected, but HOS2 overexpression significantly suppressed the stimulated MMP13 expression at both RNA and protein levels. This is for slide 13, I think. To examine if the suppressive effect of hyaluronan is dependent or not, chondrocytes were pre-treated with different doses of doxycycline before treatment with IL-1 beta. As a result, MMP13 was strongly suppressed in proportion to increases of doxycycline concentration. So usually you would say that MMP13 was suppressed in a dose-dependent manner of doxycycline concentration. So, and that pretty much says what you're trying to say is that an increase in doxycycline concentration, as the doxycycline concentration increases, MMP13 suppression was also increased. So they are directly correlated. So I would probably say it as a dose-dependent response. So in the same, this is for slide 14. In the same experiments, hyaluronan levels within the paracellular matrix and conditioned media were examined by HA ELISA and agarose gel assay. As expected, a pronounced increase in hyaluronan was observed in proportion to doxycycline concentration, and all of the hyaluronan produced by the MYC-HOS2 transgene were high molecular weight. 15. To examine the effect of HOS2 overexpression on the retention of aggrecan, primary transduced chondrocytes were cultured in 3D alginate beads. In this case, the chondrocytes were transduced with an adenovirus with ZS green HOS2. So these can be seen with fluorescent microscopy. Without IL-1 beta treatment, HOS2 overexpressed beads showed stronger saffron and O staining compared to control beads. More importantly, after 3 days treatment of IL-1 beta, conditions in which most of the saffron and O staining was lost, HOS2 overexpressed chondrocytes showed a strong resistance to IL-1 beta induced proteoglycan loss. Slide 16. The effects of HOS2 overexpression were not limited to MMP13 activation by IL-1 beta. HOS2 overexpression reduced MMP13 protein production in human chondrocytes activated by TNF-alpha, lipopolysaccharides, or hyaluronan oligosaccharides. Okay, so now for a little bit of discussion. So several mechanisms may be responsible for the suppressive effects of HOS2 overexpression. First, the most straightforward mechanism is the physical effects of secreting large amounts of newly synthesized high molecular weight hyaluronan. This hyaluronan provides enhanced retention of aggrecan proteoglycan. High molecular weight hyaluronan and bound proteoglycan may physically block the binding of IL-1 beta and its receptor. Second, the release of large amounts of hyaluronan could displace or block the binding of low molecular weight hyaluronan or other DAMPs to TLR4, as has been previously proposed by some investigators. Lastly, the overexpression of HOS2 may induce intracellular changes in transduced chondrocytes, such as affecting regulatory signaling via O-linked acetylglucuronidase. Is that glucuronidase? Sorry, that's O-linked acetylglucosamine. So I'll say that last part again. And lastly, the overexpression of HOS2 may induce intracellular changes in transduced chondrocytes, such as affecting regulatory signaling via O-linked acetylglucosamine transferase, or O-glutanac transferase. So in conclusion, the overexpression of HOS2 inhibits MMP13 production in human osteoarthritic chondrocytes, and hyaluronan increased by HOS2 overexpression could be responsible for the suppressive effects of MMP13 expression. So to summarize, we saw pronounced increases in newly synthesized HA was observed in proportion to an increase in HOS2 overexpression. The hyaluronan synthesized was of high molecular weight. Activated MMP13 expression was suppressed by HOS2 overexpression. HOS2 overexpression did not reverse the depression in anabolic gene expression, and chondrocytes overexpressing HOS2 showed resistance to IL-1 beta-induced proteoglycan loss. In conclusion, thank you for your time and all that wonderful stuff. So I tried to pause in between slides so I wasn't using a lot of time for uhs and ands, and that took about 10 minutes. So I don't know what part you want to get rid of, but you have a lot of results slides, and maybe can combine the ones where it showed that you were successful increasing your hyaluronan and then its effects on MMP13. Like if you can have that small in the corner and say like, you know, here we prove that we increase our hyaluronan amount, and not spend so much time on that. But it took me about 10 minutes to get through, so you might have a little bit more than you need for 6 minutes. I hope that helps. Sorry I couldn't spend more time on it. I actually have 4 exams coming up this week. I have 2 on Monday, 1 on Wednesday, and 1 on Thursday. So sorry I couldn't spend more time on it, but you better send me some baby pictures. And I look forward to when I can come visit you guys in Japan, and if you ever end up in Greenville, you better give me a call. All right, Shinya, Naoko, I miss you. I love you guys. I hope you're doing well, and I hope to see you soon. Good luck on your presentation.